All right, hello everyone, and welcome to the Exam 1 review for Anatomy and Physiology 1. So first here, just to go over some groundwork on the exam and an overview of the exam. So remember, this is on chapters 1 through 4. Sorry, so that's the intro, that's the chemistry, that's all stuff about the cell, and then it's the tissues. So now on this one, I have multiple choice, true and false, matching, and that's all. So remember, there is the practical then. The practical is fill in the blank where you write the word. I'll talk a little bit about the practical first here. So the practical, there are 21 questions planned. So I graded out of 20. And the way that works is you rotate around different stations, and each station will have a note card asking a question. Something might be numbered, something like that. So this first section here, I can ask things like, what sort of section is this? Is this the proximal or distal end? Is this gross or microscopic anatomy? Little things like that for this first practical. Another big part of this first practical is looking through the microscope and being able to identify certain tissues. And hopefully you looked at all the tissues in the lab and we can identify them. So for studying for the practical, you want to look over the virtual labs and also look over some of those Kahoots we played maybe as well, especially if you're looking at the tissue section. I think I'm putting like 8 tissues or so on the practical. So yeah, that's a bigger part of the practical. So now the exam itself, again, 90 questions. I graded out of 85, so that's 5 bonus questions on it. So not horrible, and you should be done, reasonable amount of time. It won't take the entire class. All right, so first here is chapter 1. This was the summary, the introduction to anatomy and physiology. So starting out with this one is, what is physiology? What is anatomy? Make sure you can define those things. The difference between studying large body structures visible to the naked eye, so gross anatomy, compared to studying small stuff, which is microscopic anatomy. Understanding the definition of what a tissue is. So, tissue compared to the organ, so different levels of organization there. And then the next big part of this one are, anatomical directions. So directions, and I'll also write regions here too. So both anatomical directions and regions. I know my handwriting's not great, but you know what I'm saying. The captions down there should pick it up just fine. So anatomical directions: proximal, distal, superior, inferior, caudal, cranial, medial, lateral. Be able to define body regions, parts of the body using these terms. You'll see these questions appear both on the practical and on the exam. So that's a big part. This should be part of your terminology now. With the anatomical directions and regions, you also have the different planes. So you have a sagittal plane, you have a frontal or coronal plane, and then you also have a horizontal or transverse plane. So that'll be big ones. You'll see questions on that stuff there. And then in this chapter, we also talked about different feedback mechanisms. So remember, we have, that seesaw that let me draw this a little better here. The little homeostasis seesaw, where, it could go up on this side, come back down on this side, then you have the part that goes up through here that's regulating it. So over here, you would have a stimulus, you would have the affector here, and a receptor and an effector. You have the control center, and then you go back out the effector to an effector organ, and you bring it back. So if it's something like temperature, you have temperature sensors, which is the stimulus is the temperature change, and then the receptor in your skin detects that temperature change, sends a pathway to the control center, says, oh, look, it's hot outside, sends an effector out to your sweat glands, you initiate sweating, you begin sweating, and it brings the seesaw back to homeostasis. So make sure you understand the different parts of the seesaw. You might see it again. And understand what homeostasis is. So it's all about keeping a relatively stable internal environment within limits. Different cavities and different region-wise. So understand what the thoracic cavity is, and then understand what the vertebral cavity is. You have your dorsal cavity and the different parts in that thoracic cavity and what you could find in it and so forth. Another big part to this first chapter here are all the systems. So make sure you understand and can define each system. So the respiratory system, the nervous system, the skeletal system, muscular, cardiovascular, integumentary, endocrine, lymphatic, and immune systems. So, lots of systems. And it's not too bad. It should be, easier to define each of the systems. They should be easy questions for you. So that's chapter 1, the little intro stuff. The big thing there is getting those terms down. And then we move into chapter 2 here. So chapter 2 was the chemistry of life. So the important parts of all the chemistry. So this, includes the macromolecules. So, we have DNA, nucleic acids. DNA, nucleic acids. You have DNA, you have RNA. And they're all made of those nucleotides. Then we have proteins. Remember, an enzyme is a protein. An enzyme acts to lower the activation energy of a reaction. And, proteins are made up of subunits. The monomers in a protein are the amino acids. And then we have lipids. So understand the different classes of lipids. You don't have to draw any or memorize the structure or anything like that. Just know if you see a lipid that it's a lipid. You're looking for hydrocarbon chains. And then the last one are the carbohydrates. So that's like our glucose, our sucrose, lactose, maltose, things like that. And remember, they are termed monosaccharide for a single sugar. That's the monomer. 2 sugars bonded together are a disaccharide. And then more than 2 sugars is termed a polysaccharide. All of these macromolecules can be made and broken. Making these molecules are a dehydration reaction. Adding monomers together and forming a polymer is dehydration. So water is removed in that process. Then you can break them apart. So what making them is catabolism. Did I say making? I meant breaking. So catabolism is breaking them down. And this is breaking them down is known as hydrolysis. So literally means water cutting, using water to cut these bonds. And then anabolism is making. So this is a dehydration reaction. At least in our body. Remember, there are the big generic terms of synthesis and decomposition reactions, and there are exchange reactions which do a little bit of both. All right, a little bit of an aside there for macromolecules. Back to basic chemistry and like overall terms. So what is a suspension, a colloid, and a solution? So understand those. And looking at the atomic model, what is the proton? If an atom has the same number of protons and different number of neutrons, what is that known as? So no, that's an isotope. And then, if the atomic number, so if, the big number on the periodic table is 11, that element is whatever 11 is. If that proton number changes, the element changes. So only neutrons and electrons can change their numbers. And then, what else do we have here in this section? Oh, it's the different bonds. So we have covalent bonds. We have ionic bonds. So an ionic bond is a transfer of electrons, causing a charged attraction between 2 elements or 2 atoms. Those atoms are called ions. A positively charged ion is called a cation. A negatively charged ion is called an anion. And then you can have covalent bonds. Covalent bonds is an equal sharing of, if it's an equal sharing of electrons, it's termed a nonpolar covalent bond. Remember, we have the Octet Rule. So the Octet Rule is when an atom wants to have 8 electrons in its outer shell. Then, with that, you can form both polar and nonpolar. Hold on, let me write some of this down. So bonds, they can be ionic, they can be covalent. And covalent can be nonpolar, which means there's an equal sharing of electrons. There's no sidedness to it, or they can be polar. The biggest polar molecule here is water. So oxygen is very electronegative, so it has a partial negative charge on it. Hydrogen here is a partial positive. So because of this, we can form hydrogen bonds with water. So if we were to draw another water molecule down here, we would draw the hydrogens right here, and then down here to the oxygen, another hydrogen there. We have the partial positive here, partial negative here, partial positive here. Opposite charges attract. Now, this isn't as strong as an ionic bond. So we draw a little dotted line right here. I know it's hard to see. This represents a hydrogen bond. But make sure you understand what a hydrogen bond is, because they are important. They're kind of an essential part to life. This is how water can dissolve so much stuff, all the ions that move through our blood, all the proteins that move through our blood. It makes things soluble. So also, with this, hydrogen bonds are the weakest between ionic, polar covalent, and nonpolar covalent bonds. If you're comparing all of them, the weakest bonds are hydrogen bonds. So if you think of a protein, so a protein, again, is a chain of amino acids, and they have different R groups coming off of it. So those amino acids have these R groups. These R groups can vary. They can be polar, they can be nonpolar, they can be charged, so they can form ionic interactions and so forth. When this protein folds together, let's say this protein folds, let's see, 1, 2, 3, 4. I'm making this up as we go. We have 5 molecules here. Let's say that one folds back into there. This is how we fold. Let's say this R group forms a hydrogen bond right there. And this one, no, I didn't draw this right. Let's just say this one is a positive and this is a negative now. So in this case, if we were to heat this mixture up, the first bond that would form here would be, or that not would form. So when you heat something up, what happens is you denature the protein. So you denature. So pH, temperature, salt concentrations can take this protein out of its natural state. The first bonds to break are these hydrogen bonds. So if you were to heat this up just a little bit, the hydrogen bond could break, and now you still might have this charge interaction right here for this ionic bond between that positive and negative part here. But now this might turn into this. Let's see if I draw this right, and then this one goes up, and now this one comes back here. So now this is a negative, and that's the positive ionic bond. But this protein now denatured into this because this hydrogen bond broke. And that's just, heating it up a little bit. And this is something as simple as a fever. So a fever could lead to a slight denaturing and breaking of these hydrogen bonds in these enzymes. So what else about chemistry here do I need to mention? I think we got most of it. Another popular protein found in the body that we talked about in the tissue chapter, and we'll talk about again, is collagen. So collagen is the one that provides high tensile strength, and you usually find collagen in tendons. I'm just looking over my notes here, making sure I'm getting it all. So, like glucose, we talked about carbohydrates. Glucose is an example of a monosaccharide. So it is one sugar. Oh, and one thing I didn't mention with the last chapter for the introduction, we also talked about the serous membranes. So understand the different regions of the serous membrane. So you have the one that lines the actual organ. So the one that lines the organ is called the visceral pericardium, if it's lining the heart. And then the one that is actually on the cavity wall is called the parietal pericardium. So that's important to understand. And in this chapter, we also talked about, now back to chapter 2 here, pH. So understand pH and understand the lower the pH, the higher the hydrogen ion concentration. The higher the pH, the lower the hydrogen ion concentration. And here, if I were to draw this amino acid again, let's say, not this one, let's draw a new one. So this is also known as a dipeptide. So we talked about, a disaccharide, meaning 2 sugars. You can say the same thing for a dipeptide. So a dipeptide would be 2 amino acids. So this would still be a protein. And if you had a dipeptide and you wanted to break this peptide bond, so this is called the peptide bond between 2 amino acids, if you want to break this, you undergo hydrolysis, not dehydration. So then, hydrolysis, water being used in this reaction, via an enzyme. In this case, it's a protease, and then you can break it into its individual amino acids then. Sorry, I know that was a bit of an aside, but I didn't want to forget to talk about pH there. And next we move into the cell chapter. In the cell chapter, first, we looked at the big overview pictures of the cell for the cell membrane, the different components of the cell membrane, why the cell membrane is the way it is, and so forth. So cells can engulf things, they bring things in different ways. So there's phagocytosis, pinocytosis, receptor-mediated cytosis. Hold on, I should keep writing here instead of just ranting. So here, chapter 3 is, the cell. So yeah, and the receptor-mediated endocytosis and pinocytosis and just phagocytosis are the different examples of how things could enter the cell. But the big part for this one is the plasma membrane. So understand the different things that can embed themselves in the plasma membrane and what the plasma membrane is. So it's a phospholipid bilayer. So here, these are phospholipids. Phospholipids have a polar head and non-polar tails, and that's why a cell is able to self-assemble itself into its own structure here. You can also have a cholesterol ring, and this is the chain of cholesterol embedded in these phospholipid membranes. So this would be cholesterol. Other things you can have embedded in here are proteins. So if I were to draw a protein, there could be one on the outside, and this would be known as a peripheral protein. Some could be embedded in or even all the way through the membrane with a channel. Then over here we would have more phospholipids. I know my phospholipids are getting worse as we're going here, but this would be an integral protein. An integral protein can also just go partway in as well. It can just go to there, and then it could be another phospholipids up there. And these are the last ones I'm going to draw on this side because we're running out of space over there. So there, integral protein. Sorry if you just heard my dog sneeze in the background. Yes, I am recording this from home for you guys. But integral protein, this can be one too. And then what else can we find in the cell membrane? You can have things sticking out. So if it's connected to the phospholipid here and is a chain of carbohydrates, let's say we have a chain of carbohydrates, down here's another phospholipid. This is called a glyco, because it's carbohydrate attached to a lipid. And then let's add another phospholipid. And now let's add a protein in here. It might go all the way through, but let's just draw it to there with another carbohydrate chain attached to it. I know it doesn't look perfect, but this one is now called a glycoprotein because it's attached to a protein. And you have a good representation of this in your workbook and stuff that you can look at as well. So make sure you understand that. Then make sure you look at tonicity. So hypertonic, hyper, hypo, and iso. So hypertonic, hypotonic, and isotonic. Isotonic is no net change. That's what you want your red blood cells in. So if you place a red blood cell in pure water, if it's pure water, where's water going to go in that blood cell? Pretend you are the blood cell. So it is a hypotonic solution. So in a hypotonic solution, you will swell and burst. So the water will actually come into the blood cell to try to decrease the concentration inside, and the red blood cell will burst. Whereas if it's a hypertonic solution, the concentration outside is much higher of solute, so water will exit the cell trying to lower that concentration, and the cell would shrivel. So shrivel, swell, and then iso is no net change. But make sure you understand what could happen to a red blood cell under these different conditions, because they're important for IVs and whatnot. And then different components of the cell. So like what a microvilli is, that's where there are projections coming out of the cell to aid in a higher surface area for more absorption of nutrients. Understand diffusion. We talked about all different kinds of diffusion. So remember, it's all about the greater the concentration gradient, the faster the rate of diffusion. So here, diffusion, I'll write that down. So everything wants to go from high concentration. Remember, brackets mean concentration, high to low. And also, they want to go to their opposite charge. So then you can go from positive charge to negative charge. Opposite charges attract. So this is called the chemical gradient. This is called the electrical gradient. And together, it's the electrochemical gradient. That's what that means. It's a combination of both. And so we talked about the importance of this in class and how we build it, and the difference between active transport requires ATP pushing something against its concentration gradient compared to passive transport where something's moving down its concentration gradient where it naturally wants to be. And then so there's primary active transport, that's like the sodium potassium pump, pumping 3 sodiums out, bringing 2 potassiums back in. And then there's something like secondary active transport, which uses that concentration built by primary active transport to bring it and something else in. A good example of this is, so if we draw a cell membrane right here, draw a protein right here, over here is the sodium potassium pump. So 3 sodiums out, 2 potassiums in, and this uses ATP. That becomes ADP in this process. So we're building a high concentration of sodiums out here. Sodiums, lots of sodiums out here, less sodium inside. So bracket, low concentration of sodium. This takes ATP in order to do this. Now let's imagine we're in our small intestine and we have something like glucose that needs to come in. And there's this symporter here, which means it can transport 2 things. Glucose has to come through this, but the only way it can come through this is if sodium comes through with it. So sodium has to come through, and as it does, it brings glucose in. Sodium is coming down its concentration gradient, so it wants to do this naturally. So then sodium and glucose, here, I'll just draw it as a circle here. That's a glucose molecule, trust me. So sodium and glucose both come in, and this is secondary active transport. This will not work unless you build this sodium gradient on the outside. So this is all about transporting a solute using the concentration or electrical gradient that's already established. So then we have the different junctions. So we have tight junctions, we have desmosomes, which are the anchoring junctions, and then we have gap junctions. So cells that are communicating with each other, sending ions to each other, like cardiac cells in the heart, have lots of gap junctions. Cells that have a great deal of mechanical stress and stretch, like intestine and bladder, we see a lot of desmosomes. And then tight junctions are areas where you need a lot more seal between the cells, and you want to hold more of those cells together. And then I talked about the different kinds of transport, bringing stuff into the cell and out of the cell. So endocytosis is entering the cell, exocytosis is exiting the cell. And then know the organelles. So know the different endoplasmic reticulums. There's rough ER, smooth ER. There's the Golgi apparatus. There's the vesicles. You have a diagram of a cell. Make sure you understand that in all the parts of the cell. It should be second nature by now, but cell. I'll just write cell, know all the different parts. And then a little bit about the DNA. So DNA, remember, is a sequence of nucleotides, and that codes for genes, codes for proteins. So a gene is a segment DNA that carries the instructions for one polypeptide chain. A polypeptide chain is a protein. Energy currency of our cells, everyone should know this. ATP. That's adenosine triphosphate. And then if we have a DP, how many phosphates do we have? This is diphosphate, so that's 2. AMP is 1. I'll draw a 3 here for the T. So AMP is adenosine phosphate. And then we did talk a little bit about the cell cycle. So here's cycle. Oops. So cell cycle, know the different phases. I'm not going to go into crazy detail in this, but understand prophase, metaphase, anaphase, and telophase, pretty much. And that this cycle is controlled, and any lack in that control could lead to tumor growth in something that's cancerous. So for proteins, if you find a cell that has a lot of, rough ER, that's the protein factory, or protein factory for proteins that are being secreted. So a lot of rough ER and a lot of Golgi apparatus. What is something we expect about that cell? It's probably going to be secreting a lot of proteins, maybe something like insulin or even mucin, like we talked about in the tissue chapter. Oh, one other thing I didn't label up here. What are the different regions? So this is the polar region of the lipid, and this is non-polar. The cell. And then we get to the best chapter here. At least my favorite chapter of this unit, chapter 4, tissues. So we have the major tissue types. And yes, we have to know them. One for the practical and then also for the lecture portion of the exam. Doing the virtual lab, checking out the Kahoot is the best way to figure this out. There might be similar questions, especially from the virtual lab. So doing that multiple times, looking at the different parts there and seeing it, understanding what each tissue type is and what it looks like in the microscope is important. So I'll just do a quick little overview here. So, if for doing histology, histology is the study of tissues. And then to prepare these slides, they have to be fixed. So using a fixative such as formaldehyde. Then they have to be sectioned, which means they have to be cut very, very thin. And then they have to be placed on a slide and then differentially stained so we can see them. And then we can see the pretty pink and blue colors for the nuclei. So all the different tissue types, starting with the epithelial tissue. So epithelial, it can be simple, which is one layer, or it can be stratified. Then each one can have 3 options to it. So simple can be squamous. Simple can be cuboidal. And simple can be columnar. Stratified, so simple is one layer. Squamous is flattened. Cuboidal is cube-like. Columnar is column-like. Stratified then can be also squamous, cuboidal, or columnar. So talking about some of these here, simple squamous, those are the ones you find in like the air walls of the lungs and the kidneys. Simple cuboidal, you can find these ones in some secretory glands and in kidney tubules as well. Simple columnar, the linings of tracts and things like that. And with these simple columnar, you can also have what are called pseudo-stratified. Pseudo-stratified means false layered. And these ones, so say we have a pseudo-stratified columnar layer and it's ciliated. And this is found in certain places. And a lot of times you find these pseudo-stratified columnar epithelium with mucus-secreting goblet cells. So those are the ones that line the respiratory tract. The respiratory tract is producing mucus, and it's lined with these cilia to get the mucus out, to get the debris out of your lungs. So little examples like that. So if you find a single layer of epithelium consisting of cube-shaped cells, be able to name that. Be able to nail that simple cuboidal. And then there's also the transitional, which is a stratified tissue. And it's kind of a mixture of all of them. It changes shape. It starts as one and goes and becomes another. Then we get down into the connective tissue. Connective tissue Has a bunch of different tissues in it. First one is CT proper. CT proper can be loose or dense. First one is loose. Loose can be areolar. See if I can name all these without checking my notes. Areolar, adipose, and the last one is elastic. Or is it reticular? It's reticular. I believe. And then we have dense. Yeah. Dense can be regular, irregular, and then here is elastic. Elastic, you're looking for these dark fibers in it. So this is like the aorta, the wall of the aorta where you have lots of stretch. Here, let me write this better here. Elastic. Irregular is a bunch of tissue going different directions. So pull from opposite directions. Where regular, it's all going the same directions, like in our tendons. For loose, areolar, this is made up of different fibers, and it has a lot of areolar space in it too. So a lot of like space as in like, water or just, ground substance. Adipose, these are fat droplets, so you're looking for little droplets like this. Reticular, then, this is what we find in lymph nodes and spleens, and we have these large reticular spider-like fibers in it. And then you have cells all over in here. You can see the space and so forth, and you're looking for these dark reticular fibers. That's CT proper, loose versus dense. And then the next type of connective tissue are the cartilage. So remember, what you want to look for in the cartilage are those lacunae, or the pockets. So three main types of cartilage. We have hyaline cartilage, we have elastic cartilage, and we have fibrocartilage. So again, the key is you're looking for the pockets of lacunae. Hyaline is the most dense of the types of cartilage. It's found in our coastal cartilages of our ribs and so forth. I could draw it, but it's hard to draw it in just one color here. So there's the pockets, and then the ground substance is equal throughout the whole thing. So like I said, it's hard to draw that one. The elastic one, you have the pockets of lacunae, but then you can see these dark fibers in it as well. And that's the elastic pool like we find in our ears. And then the fibrocartilage, this is compressive cartilage. And these fibers in the elastic ones might be going different directions as well. And then the fibrocartilage, we find this in not our ears. Did I say ears? So this is found in our vertebral disc and meniscus of our knee and our pubic symphysis. So this one, you're having fibers, you have lacunae in there, and then they're all running the same direction. So imagine a compressive shock pushing down here. These collagen fibers are going to resist that compressive shock. So when you're trying to figure out, is it connective tissue proper or cartilage, you want to look for these pockets of lacunae. And then that's the key thing here, because some that are easy to screw up are fibrocartilage versus things like reticular or regular connective tissue. So you don't want to make that mistake. Easy mistake to make too. Another type of connective tissue is bone. Remember, whenever you see osteo, think bone. And we have the different cells. We have osteocytes. Let's start with osteoblasts. And they become osteocytes. So osteoblasts are young osseous tissue forming cells. And then the osteocytes are the older, mature ones, which are maintaining the region. If the cartilage, we have chondroblasts and cytes. And then up here in the connective tissue, we have fibroblasts and cytes for the most part. Adipose has adipocytes is what they're called. But these are fiber producing cells. So I wanted to get the cell names out there. So for bone is easy to identify. You have the rings. And we'll talk about those rings more when we get to the bone chapter coming up really soon. And then blood. Blood is considered a connective tissue, so you can't forget about blood. You have the red blood cells and the white blood cells throughout it, and the plasma is the ground substance. Then you have nervous tissue. So you're looking for neurons. You have a little cell body here, and then these little appendages coming out of the neuron, and then one big axon then coming out of that. And these little dendrites can have little appendages coming off them, and they're all sensing the environments and so forth. But there's one big axon then that comes out of that neuron. And all neurons do look different depending on where you're looking at them. And then from the nervous end, we have muscle. 3 types of muscle. We have skeletal, we have cardiac, and we have smooth. Skeletal, voluntary, the only voluntary one, you control it, and it's also striated. Cardiac, non-voluntary, lacks striations. No, doesn't lack striations. So let me repeat this one. Cardiac is non-voluntary, it has striations, and it has intercalated discs where we have gap junctions between the cells. Smooth muscles, non-voluntary, and lack striations. I got ahead of myself a little bit there. It's the key thing with the cardiac, intercalated discs. If you ever hear intercalated discs, think cardiac muscle. And then what else do we have here? So the muscle, the bone, the blood, I think, and then we have, I think we're close. I think I'm just kind of talking here about all the tissues. I think we're close to getting them all. Don't forget, you have the membranes. The cutaneous membrane is the skin. You have the serous membranes, and then you have the the linings, the endothelial membranes and things like that. So you need to understand those terms. And we have the glands too. We can't forget about the glands. So if they secrete their products like directly into the blood, so those part of the epithelial are the glands. Remember, if they secrete directly into the blood, they're endocrine glands. If they secrete to outside the body, they're termed exocrine glands. See what other questions we could have here. The main difference between hyaline cartilage from elastic and fibrocartilage is that the fibers are not normally visible for the hyaline up here. Usually you can't, I know I drew these fibers in here, but usually you can't see those fibers. So ignore that structure. I was trying to draw the ground substance outside. So here, structure, remember when we looked at the goblet cell, they have a lot of Golgi in them because they're secreting vesicles of mucin. I think you guys will do well on this. If you understood most things I was talking about in this review, you should do okay. Oh, and the last thing we did talk about tissue repair. So understand the tissue repair is all about inflammation, regenerating, restoring a blood supply, and then regenerating, and fibrosis, which fibrosis could have the different scar tissues, and different cells can regenerate at different rates. Well, not different cells, different tissue types regenerate at different rates. So, have a rough understanding of that. I might not ask any direct questions, but still, it's important to understand that, cardiac cells and neurons, they don't regenerate. No regenerative capability. Yeah, I think this is a good overview. I'm just scanning through my notes here, making sure I hit everything that should help you guys. Just making sure I didn't forget anything. I think we're looking good. And let's see, that's just a couple things. Remember, the fibrocartilage is about absorbing those shocks. Oh, I don't know. Back here for this, the glycolipid and glycoprotein together are known as the Glycocalyx. Forgot to mention that. I meant to mention that there. So if you see Glycocalyx is just a combination of glycolipid and glycoprotein. Oh, and back to the cell. So the central dogma goes DNA to RNA. So I guess that would be down in here. So DNA to RNA, that's transcription. And then RNA to protein, which is translation. Oh, and a few other things about the epithelial cells. Do they contain blood vessels? No. They are avascular. They are innervated, though, which means they contain nerve fibers. So they do not contain blood vessels. And blood, again, is considered a connective tissue. But yeah, I think that is a good one. So other things you guys should do besides looking at this video and highlighting the stuff that you might have, forgot or like, oh, wow, I don't know any of that. Make sure to review that. Review the lecture quizzes. So chapters 1 through 4, and then review the virtual labs and your workbook. If you do all that, you should do fine on the exam and the practical, but you have to study, you have to work on it, you have to study it. If you have any questions, feel free to email me, and I'll get back to you as soon as I can. With that, I hope you have a great day, and good luck.